Good day, everyone. I am Nirmala Ganessingh, acting agricultural entomologist at the Research Division, Ministry of Agriculture, Land and Fisheries. Today, my presentation will be on an insect pest, a psyllid called Chrysaenus latapenis on Pommecythere, identification and management options. So you may be asking yourself, what kind of pest is this? Chrysaenus latapenis is an insect, a psyllid. Psyllids or jumping plant lice are characterized by the very narrow host plant range. It originated in Polynesia, which is South Pacific, identified in Malaysia, Thailand, Vietnam, and recently in French Guiana in South America in 2021. The wide distribution of this insect in Asia suggests that the species has the potential to become invasive in the neotropics wherever the host plant occurs. For your information, invasive means spreads quickly and is harmful, causing damage. Neotropics is the tropical New World, including Central America, South America, and the Caribbean. Last year in April 2021, this insect, Chrysaenus latapenis, was confirmed in Trinidad at Farm Road, Carib. It was then identified in other locations in Trinidad: Kelly Village, County Caroni, La Reunion, St. George East, Barapore, County Victoria. The picture on the slide shows Pommecythere seedling plants infected with insect pests. The host plant for this pest is Pommecythere, also known as golden apple. Pommecythere trees in Trinidad and Tobago are found scattered throughout the country, growing in backyards and roadsides with little or no agronomic attention being paid to them. Pommecythere fruits of both genetic lines, the dwarf and regular varieties, as you know, can be utilized in the fresh and processed forms. This slide shows the utilization or usage of the Pommecythere fruits. Small orchards of Pommecythere have been established by farmers and agro-processors in Trinidad for fresh fruit usage and processed fruit usage. Fresh fruit usage includes for the export market, where large fruits are exported, for the food caterers, like example showers and chutneys, processed food utilization as value-added products, preservatives, pickled and confectionaries, fruit juice and concentrates, frozen pre-cut Pommecythere, and beverage wine industry. This slide shows both the dwarf and the regular large tree-type varieties of Pommecythere that are susceptible to this insect. The psyllid feeding and behavior on the host plant Pommecythere. The insect feeds by sucking the sap of the host plant, in this case Pommecythere, which results in poor plant growth and decrease in fruit production and yield. So this insect is a sucking insect sucking the sap of the plants. So how this insect spreads? Psyllids are inadvertently or unintentionally introduced to new areas along with their host plants. These tiny insects have hind legs that allow them to jump and fly away quickly if disturbed. Psyllids can spread through the movement of infested plants, planting material, nursery stock, and machinery and equipment. So, if there is trade or movement, for example, through the sale of infected plant material from one location to the next, this pest would spread, as the infested plant material will contain the insect eggs, nymphs, and adults. This slide shows the pest life cycle. The life cycle includes egg, 5 Nymphal Instars, or called immatures, and adults. So if the plant is affected by this pest, the management strategies would have to include controlling stages of the pest life cycle. This picture shows the psyllid eggs and what they look like. If you look at the photo, they are yellow, oval, and the tips orange in color. This slide shows immatures or the Nymphal Instars. There are 5 Nymphal Instars in this psyllid. They are yellowish with red eyes. This picture shows the adult psyllid. The adult is 2 millimeters to 3 millimeters in length and black. Iridescent in color meaning shiny. So how do we identify this pest on the host plant Pommecythere? How do we do that? What damage and symptoms are observed on the plant? The most notable symptoms are stunted shoots with densely curled leaves, as seen in the photo. Identification continuing the damage and symptoms observed on Pommecythere plants. You would observe white waxy secretions on curled leaves as shown in the photo. I believe you are seeing the photo now. You would observe white waxy secretions on curled leaves as shown in the photo. Continuing on symptom damage seen on Pommecythere plants. Psyllid immatures develop in small colonies on Pommecythere leaves tips and open or closed twisted leaf rolls or galls. Right, so those are the symptoms you'd be seeing to detect if you have this insect pest. Under magnification, you would observe all stages of the insect life cycle happening at the same time. You would see the adults, eggs, and immatures. Only using a microscope you'll be able to see this insect because the insect is very tiny. So the picture shows eggs and Nymphal Instars, and you can see this under a microscope. This slide you see in shows the psyllid damage to both dwarf and the large regular tree-type varieties of Pommecythere plants. Right, the damage observed at the tip of the plant or growing points, as shown by the red arrows. So field observations and early insect pest detection is important. Take time to observe your fields for pests. Field observations allow the grower to monitor crop growth and to notice if there is a presence of pests or diseases that may result in yield loss. Timely identification and early detection of pests and disease in the field decreases crop yield losses and economic losses. It enables the farmers to understand pest and disease activity, progression, permitting early adoption of cost-effective and environmentally sound pest management decisions. The picture shows protective layer. So how do we manage it? How do we manage and control this psyllid? This slide shows the management strategies of the psyllid on dwarf varieties and seedling plants of Pommecythere. So these are the smaller plants we're talking about here. Number 1, remove affected plant parts. Prune and dispose the affected foliage by burning where possible. Number 2, apply insecticide using a knapsack sprayer. Apply systemic insecticide, for example, acetamiprid, active ingredient, used as a foliar spray, meaning sprayed onto the leaves or foliage of the plants at manufacturers' recommended rates and intervals. Or apply systemic insecticide, for example, imidacloprid, active ingredient, as a root wrench, applied meaning it is applied to the roots of the plants and it is absorbed or soil at manufacturers' rate and intervals. 3. Fertilize. Apply growth promoters and bud boosters. These enhance plant growth, root and flower node development. Apply granular fertilizer as 12-12-17-2 or 13-13-21 as required. Maintain field sanitation, number 4. And number 5, encourage natural enemies. For those who don't know what a systemic insecticide is a systemic insecticide is an insecticide that plants can absorb from the soil via roots as a root wrench or through the leaves as a foliar spray, and it is then spread throughout the entire plant, reaching stems, leaves, flowers, and fruits. Remember to always wear protective gear when applying insecticides. This slide shows management strategies for the psyllid on large regular tree-type varieties of the Pommecythere plants. So these are large adult trees we're talking about now. Number 1, remove affected plant parts. Prune and remove from the field affected plant parts. Burn material away from the field. Number 2, apply insecticide using a mist blower. Apply systemic insecticide, example acetamiprid, active ingredient, as a foliar spray. Alternate with systemic insecticide, for example, spirotetramat, active ingredient, according to manufacturer's rate and intervals. Number 3, fertilize with 12-12-17-2 and calcium nitrate at a ratio of 1:1 at the onset of rainy season and again at October-November. Number 4, maintain field sanitation. Control the weeds in the field. And number 5, control natural enemies. Again, wear protective gear when applying pesticides. A field survey on this psyllid is going to be conducted by Research Division. The survey will cover all 8 counties in Trinidad, and the survey will be conducted in collaboration with RAN and RAS and other stakeholders. The survey is to be taken to Tobago after its completion in Trinidad. So you may be asking yourself, why field surveys are important to undertake? Surveys allows for features of an area to be protected or to adopt appropriate levels of mitigation measures. Mitigation measures means action taken to reduce severity. That's me in the photo along with my colleague, Mr. Mukesh Ramdani, Agricultural Officer 1. So the expected outputs from the survey: to determine the pest population spread in Trinidad, meaning pest presence or absence, to illustrate and report on pest-free areas, if any, to provide pest incidence and distribution information, to assist in determining management strategy for the pest, to determine if there is alternative host plants, meaning that if we find this insect feeding on other plants or other species, to promote transparency in trade, thereby increasing confidence with trading partners. Elaborate further is allow for ease of export of the crop from Trinidad and Tobago. Data collection from a survey facilitates confidence in trade. The exporting country that conducts a survey is able to forward and share that data from the survey and the protocol used, which increases the country's ability and chances to trade with another. Right, so those are the reasons why we would want to be conducting a survey on this particular psyllid. So we encourage you at this point to contact us if you suspect you have this pest. Our office is available to visit you at your farm, homestead, and even at your backyard garden to provide an assessment. You can also forward emails with pictures to the email address shown, entodialaboratory@dove.tt, and we will respond and assess your situation. You can also contact us at Research Division 646-4337 or at the Ministry of Agriculture, Land and Fisheries Head Office number at 220-6253, and the information will be forwarded to the Entomology Unit Crop Protection Research Division. So this brings the end to my presentation. Thank you. I would like to extend special thanks to my colleagues at the Crop Protection Unit Research Division, in particular Mr. Terence Jock, Plant Pathologist, and Mr. Rishi Mohan Singh, Agricultural Entomologist, and lastly, Mr. Mukesh Ramdani, Agricultural Officer 1. I also would like to single out Ms. Diane Ramrup, our former Director at Research Division, now Director at ETIS, for her dedication to crop protection in Trinidad and her valuable contribution in this awareness presentation. At this time, we will address any questions pertaining to this topic. Any questions would be addressed at this time, so you can type up your questions if you have any questions concerning this topic, or this insect or psyllid. Any questions? Anybody has this insect we would want to come and have a look at? Okay. Okay, well, all right, yeah. Okay, so since I'm not seeing any questions or people shy, they could type their questions later on the day, and we will always look at it and reply thereafter. Right, so at this point, I'll bring my presentation to a close, and thank you very much for listening. And you could always contact us at the numbers provided, and you could always look at it later on to see if the numbers registered on the Facebook page. Right, so thank you and goodbye.